Good morning. Good morning, everybody. It's 5 to 11, so I'll just have one or two little introductory comments. The first is that we've got 3 boards here that I'm going to send around the room today, and those are so you can put your write your ideas for a lecture series for next year. So there are 3 boards and plenty of room. Included is a column for any ideas that you might have for new interest groups to operate within U3A Rutherford. So there's the ideas for next year's subjects and ideas for interest groups looking forward. We are still collecting wine bottle tops. Just a reminder, if you have a huge collection of them at home, you can bring them here next week. So thanks to Jacqueline Smith for organising that. So welcome everyone. Welcome also to those watching online this morning. Now, cell phones off, please. I'm very pleased to introduce our speaker for today. Our speaker is Dr. Tony Connor. We had this lecture scheduled a year ago, and COVID put an end to that, and Dr. Connor was very pleased and gracious enough to come back this year. We've asked him to talk about genetic engineering in agriculture. Genetic engineering has been a divisive subject around the world, and especially here in New Zealand, for many years. The media has reported on both sides, but emotions can run quite hot. So today, I'm sure we will learn the hows and the whys. Dr. Connor's research is mainly with plants. His research career has focused on the application of plant biotechnology and genomics to crop improvement. Much of the research has been with potatoes, but also with a range of other vegetables and forages. Tony retired in 2020 after 10 years as science crop leader for forage science and ag research, and he worked for 30 years at the Lincoln campus of crop and food research. When he retired, he was awarded the status of emeritus scientist at ag research, and that is indeed a high honour. He graduated BSc Honours from Canterbury and has a PhD in genetics from the University of California, Davis. He's a companion of the Royal Society of New Zealand, a Fellow of the Institute of Agricultural and Horticultural Sciences, and a Fellow of the Royal Society of New Zealand. Dr. Connor, you are exceedingly well qualified to help us separate the wheat from the chaff on this important issue.
Eira rangatira mā, tēnā koutou katoa, nau mai, haere mai, ko Tony Connor aho. Looks very good to be here, finally, and I'm thrilled to see that so many people are wearing masks in the audience. I think it's rather ironic that COVID is probably more common in the community now than it was when we cancelled the talk a couple of years ago, but at least now we are all vaccinated and we are protected if we get infected. And I do encourage everyone, if you haven't had your second booster yet, to get out there and get it as soon as you're eligible. It may not stop you getting COVID, but if you do get it, the impact on your health will be that much less. Now, I've got about 35 slides here. Some of them are quite quick, they're just pretty pictures. Others have text on them. They're largely to remind me what to say, but you're welcome to read them with me as well if you chose to do so. So, the menu of my talk, I was going to ask to give an overview of certain subjects, but I'm going to give you a brief introduction to genomics and molecular biology, just one slide to understand the basics of molecular biology to help you through the rest of the talk. I'm going to focus the talk around the history of genetic approaches to plant improvement, because I think it's important to understand what we've done in the past to the genetics of plants to understand what impact genetic engineering is going to have. I'm going to talk about the global status of genetic engineered crops throughout the world, where we're at the moment in terms of what is grown and where they're grown. I'm going to talk about the history of genetic engineering in New Zealand with a focus on plants, because that's where most of the work in New Zealand has been, is on the plants. I've got one slide because I was also asked to speak about animals. Now, I'm not an animal biologist, but I've got one slide that gives a brief overview of what's happened in New Zealand in animal genetic engineering. I'm going to talk about some more modern day approaches to genetic engineering that are sort of a Clayton's genetic engineering. They're genetic engineering, but they're not, but they're still legally genetic engineering. So we're going to cover that off as too, as way some of the science is moving. And I'm going to finish up with just some final reflections. Now, throughout my science career, I've been involved with genetic engineering of plants. And it's been a bit frustrating at times, some of the response to the community and society and the legal structures in New Zealand, how we've not been allowed to grow these crops. So I'll try and tone it down, but you will detect the odd level of frustration in some of these slides and some of the things I've got to say about things we weren't allowed to do. But anyway, we'll come to that. So, molecular biology and genomics 101. This is very simple. I'm sure you're all aware we have DNA, and DNA is in all the cells in our bodies and the cells of any organism. DNA encodes the information for life. And in every cell, the DNA is assembled into chromosomes, and the genes are on the chromosomes, or on the DNA. When the genes are what we term as expressed, they're turned on to have a function, what happens is that the DNA gets transcribed into RNA. That's the first step. And then the RNA gets translated into proteins. And it's the proteins that function life. It's the proteins, sometimes they're structural proteins, they form a function as a structure, but generally they are enzymes. They catalyse biochemical reactions, and that's what gives life life, is the activity of those enzymes. So those enzymes control metabolic pathways that turn particular metabolites into the next one and the next one along a pathway. So in essence, the genetic engineering is about the DNA. The DNA encodes the information, and that gets transferred to RNA and then into proteins. Now, in a standard higher organism, you might think about 30,000 genes in their genome. They're not all turned on at the same time. Sometimes genes are repeated as several copies of the same gene, and some of those genes turn on in the leaves, some in the flowers, some in the roots, and their levels of expression, as we say, may vary depending on when in the cycle of the plant it is. So not all genes are turned on all the time. So the population of RNA is always fluctuating in an organism, and every cell it changes depending on which genes are turned on. So when a gene is turned on and is made into RNA, the RNA has a life. It might be minutes, it might be hours. So it can only make the proteins at certain times in the life of that particular function. So the functions are always changing depending on the expression of the genes. Now the DNA is what we call this double helix. It's a lot of DNA in every cell. Now, I've heard it said, I haven't done this calculation myself, but I heard someone else say it. If you imagine the DNA strand being a railway track that we normally see carries our trains, there's enough DNA in every cell in the human body, if it was a railway track, to extend right around the equator of the Earth. Plants have much bigger genomes than humans. So you imagine how much DNA is in a plant cell. And it's always marvelled me right through my career, and I kind of understand the principles of how it happens, but it just amazes me that every time a cell divides, the DNA contracts back into its chromosomes, it gets replicated, then those chromosomes then separate into the two cells. Now a cell may divide when a plant's actively growing every day. And how the hell that DNA can contract back into chromosomes and separate without getting tangled up. I just find it amazing that that actually happens, so common in life across all organisms. So with that brief introduction, I'm just going to leave DNA as DNA, and you just got to know that DNA can get turned on, the different genes can turn off at different times to make proteins, and proteins control the functions in a living cell. So I'm going to quickly go through some of the history of plant breeding. Because to me this is fundamental. We've got to understand what we've done in the past to understand the impacts of genetic engineering in terms of its safety. So plant breeding really began with the human race as a human and adapting to living in communities, gathering food. So crop domestication began as soon as mankind started harvesting plants for food. And early on, in the early camps of human civilization, there was often at the camping sites waste areas and where the rubbish was thrown. And that's often where seeds ended up. And mankind soon learnt that they could harvest those seeds and plant them. But just the fact of throwing the rubbish out into a rubbish heap and harvesting food that grew from the seeds in the rubbish heap, plants soon adapted to mankind, not mankind changing plants, but the plants adapted to humans. One of the first things that happened was the loss of seed dormancy in many of our cultivated crops. But over time, mankind learnt to save seed. And as soon as you save the seed of the plants you prefer to use, you're starting breeding programs. And influence the genetics change over time. Might be very slow in those days, but it was happening early all through human civilization. Initiation of plant breeding. We had Gregor Mendel, born 200 years ago last week. In the late 1800s, he published a paper that outlined the laws of inheritance in peas as his first organism investigated. His paper lay unknown for about 20 years, but rediscovered in the early 1900s, and that stimulated a lot of an understanding of inheritance in plants, and stimulated a lot of crossing and purposeful breeding of new crops, new cultivars of crop plants. Big step forward in the 1920s with the idea that plant breeders, the early plant breeders, knew they could cross different plants. Sometimes they could cross different species to bring traits together from different species. But in the 1920s, with the advent of what we call embryo culture, you could greatly expand what you could actually hybridize. Normally when you hybridize two plants, often you can't get a hybrid. The embryo in the developing seed will abort very early on, but with the use of embryo culture, you could actually nurture that embryo back into a whole plant and then get it to form flowers and be able to, in some cases, have some fertility to do further breeding. So that was way back in the 1920s. In the 1930s, people found the role of colchicine as a chemical, as one of the first chemicals that you could double the chromosome number. It interfered with the cell division, of separation of chromosomes and cell division, so that chromosomes wouldn't separate, and you end up doubling the number of chromosomes in a plant. That was very commonly done from the 1930s to get what we call polyploid plants. Polyploid plants with double the chromosome number have larger cells. Bigger plants, more food. One of the outcomes of World War II was the use of chemicals and radiation to actually induce mutations. And these are somewhat nasty chemicals that cause mutations of DNA, and the use of radiation to cause chromosome breaks and structural changes in chromosomes. That became very common after World War II in our breeding programs of crop plants, to actually do that in a purposeful manner to induce mutations. Usually those mutations had negative effects, but occasionally you'd find the rare plant that had something positive about it. And I would say nowadays, of all our major vegetable and arable crops, every modern day cultivar will have somewhere in its pedigree some form of mutation breeding. And I can almost say every barley cultivar nowadays grown and most wheat cultivars will have radiation in their pedigree somewhere. From the 1950s, we learnt how to make haploid plants, plants with half the number of chromosomes. That was by culturing pollen, taking the pollen from plants and culturing that back into a whole plant. And because pollen has half the number of chromosomes as part of the normal meiotic breeding cycle, we're able to generate ways of making haploid plants. So now we had a system where we could double them up and halve them down intentionally. And that opened up huge opportunities for plant breeding, be able to do that. You could then take a plant that's quite variable genetically, what we call heterozygous, for many, many genes, and it's segregating, and it's not purebred. In one generation, you could halve a number of chromosomes, double it back up, and it's perfectly true breeding and homozygous for every gene. Very valuable approach to improving our plants, especially our inbred crops. Commonly done from the 1950s onwards. In the 60s to 70s, we had the concept of what we have as somatic genetics. And that is actually doing away with sexual reproduction, but changing things somatically in their somatic cycles. And down the bottom there, if I can highlight it, just this little photograph here. These are asparagus protoplasts. Now, protoplasts are plant cells that you can culture without their cell wall. The cell wall provides a structured entity for the cell to protect it. It also defines the shape of a plant cell. But if you use enzymes to digest away the cell wall in culture, they all become little green, if they're from leaves, to little green tennis balls. Very small, always round. One of the key roles of the cell wall is the cell will expand out to the cell wall under osmotic pressure. If you take off the cell wall will just burst. So in your solution, you've got to have an osmotic balance. You add often sugars that aren't metabolized by the cell or polyethylene glycol to certain concentrations to replace the cell wall to stop the cell bursting to balance the osmotic pressure. And they just round out to nice little tennis balls if they're taken from leaves. The thing about these somatic cells is that once you have protoplasts, you can fuse them with each other. In actual fact, there's no limit to fusing membranes. You can fuse often related species. I think one of the widest examples I'm aware of where you've actually fused together in the 1970s from Germany, fused together potato cells and tomato cells, fused together, and they're actually able to get those cells to replicate, regenerate, and recover plants back from them that were somatic hybrids between potatoes and tomatoes. Now, the plants were sterile. They could never be sexually reproduced, but that just shows you what could be done here. As I said, there's no limit to what you can fuse together. There is one paper in the literature that has claims to have fused carrot cells with human cells. Now, they were able to be viable and sit there and be viable, but they were never able to divide. Thank goodness. But that's a somatic approach. Another somatic approach is illustrated on the side up here. It's an example in potato. These are potato cells in culture, just growing them as single cells in a culture, much like you grow a microorganism. And what you can do with potatoes, or any plant cell, is you can plate them on a petri dish. Now, this is a petri dish. It's about 10 centimetres in diameter, just about this size here. And it's had on its surface about 100,000 plant cells plated on that surface of that petri dish. That's quite a number. If you're doing that in bacteria, you'd plate 1 million cells in a petri dish, but plant cells are much bigger. So if you can plate out several hundred of these at 100,000 cells, you've got quite a large population of cells. So what we were able to do is we exposed the suspension culture to the toxin of common scab that kills the potato cells, and then grew them out and looked for rare survivors, rare surviving cells. And we recovered some rare surviving little cell colony just there, and then regenerated a whole plant from it. And because that little cell was resistant, a spontaneous mutation for resistance to the toxin that had been exposed to, regenerate the plant, and the plants had resistance to the disease. So that's a way of somatic breeding, applying the principles of microbial genetics to plants using cell culture. That was quite a common thing. In fact, that was the kind of project I was doing for my PhD in UC Davis back in the early 80s. And during my PhD, the first transgenic plants were announced, and I think it was in December 1983, where labs from USA and Europe at the same conference both announced they'd established and provided the proof they'd established transgenic tobacco plants, tobacco being a model system for a lot of cell culture research. Now, that was immensely exciting time in plant science. It was right at the start of my career, and it was just really enthralling to be part of that community at the time. When I came back to Lincoln in 1985, I was told, you're working on transgenic plants, developing genetic engineering in our crop plants. And it was a cool time. I'll come back to this slide later to illustrate. There's a couple of little points there too. Genetic engineering has moved on. Whoops, sorry, let's go back. Get the right button. Genetic has moved on. I'll discuss briefly these ideas of cisgenics and intragenics. And then I'll also say something about gene editing, which is getting very sophisticated and targeted, what you can now do in most of our crop plants. And to a large extent, gene editing is going to take over genetic engineering as a way forward, especially now we have the genome sequence of virtually all our crop plants, and we know all the genes and all the sequence. You can now go back and you can make a change of one nucleotide on that whole genome of many, many thousands. Right, moving on. Where are we at with GM crops around the world? The best figures I could come, actually it's a couple of years old, but nevertheless it's changed over the course of time. The first GM crops were grown in the late 1990s. You might remember the Flavour Savour tomato was one of the first way back in 1996. But nowadays, GM crops are grown in about 29 countries, predominantly from North America, South America and Asia. There's routinely now, every year for several years, about 14 crops of GM status that have been grown, and they're all listed there. The big ones are soybean, maize, cotton and canola, to some extent alfalfa or lucerne. The other ones, the vegetable crops, are very minor in specific countries. The total area grown in 2019 was just over 190 million hectares of GM crops were grown globally. Now to put that area in perspective, that is more than 7 times the total area of New Zealand. And it made up 13% of the total arable land in the world were grown into GM crops. So they're there. They're not going away. These are the countries that have been growing them. You see most of the North American countries and South American countries grow GM crops now. A lot of them in Asia, a few in Africa. There have been some others who have come and gone over the years. A lot of the African countries have grown them for a few years and then they've dropped out and others have picked it up. There was a time, even in Europe, Portugal and Spain currently grow GM crops. There was a time when they were grown in Germany and France and some Eastern European countries, but they no longer grow them due to political pressures. So a couple of tables just showing you the areas, just show you where the major things are. So the big countries are the USA, Brazil, Argentina and Canada. And more recently, India is actually expanding its area of GM crops. They're the main terms of large areas. The countries with an asterisk down here have been routinely growing them for many, many years, but some of them are quite small. But notice our neighbours Australia are there with a significant area as well. And then the list continues. Again, a few of these still have the asterisks. They're quite commonly growing them. These other countries here they're sort of yes some years, no some years, come and go over time. And there's a list here of the kind of crops that are grown. And this just gives you a feel for the magnitude of different crops. The vast majority, nearly half of these GM crops, are soybeans. A lot of maize, a lot of cotton, reasonable amount of canola, mainly in Canada, and a little bit in Australia. And then all those other crops mentioned make up a very, very small total area. So, as you can see, soybeans in particular are a dominant crop. A lot of our products have soybeans in them in our food, often oils and flours from soybeans. One slide I meant to add and I forgot to was just the status in New Zealand in terms of where our food is. We don't grow any GM crops, but there have been about 30 or 40 products approved for sale in Australia and New Zealand through the Australian New Zealand Food Authority. And you can be quite sure they're on our products, on our shelves. Particularly when you see the domination of soybeans. I understand at one point in one year, 85% of the world's soybeans were GM. I'm not quite sure of the current figure. It might have come down a little bit since then. But about 2016, I think it got up to about 85% of the world's soybeans were GM. So you can be quite sure anything in your supermarket shelf, particularly those complex foods that have soy oil in them, that is a GM product. And it doesn't need to be labelled because the food is an extracted, purified product. Oils and sugars and those kind of things don't need to be labelled, only if it's a whole food. So it's probably there on our shelves. You may recall some of those cotton figures, particularly in Asia and China. A lot of the cotton is GM cotton. I'm quite sure a lot of those cotton t-shirts you see around the place, often with anti-GM slogans written on them, are probably cotton out of China. Okay, let's just go back to where we are in New Zealand. Now, lab work started in New Zealand on GM crops in the early 80s. I came back to New Zealand for my PhD in 85, and at that time there was already people in DSIR, in particular Derek White at Palmerston North was already working on white clover. And Richard Gardner, another colleague at University of Auckland, was working on kiwi fruit. And a range of labs were working on model systems, particularly what we, it sounds a nasty word, tobacco, but tobacco is a lovely plant in cell culture. It grows so easily, it's a really good model system. When I came back to Lincoln, we worked on potatoes and asparagus. But by the late 1980s, I would think all research organisations in New Zealand that were involved with plant science across the DSIR, Math Technology and the Forest Research Institute and all the universities were involved with making or developing GM plants in the laboratory, usually as a research tool, but sometimes to develop the technology so that when genes of some value came available, they could be inserted into those crops. Most of the work in our crop plants involved DSIR, Math Technology and the Forest Research Institute, and they covered, by the late 80s, all our pasture crops, arable crops, vegetable crops, fruit, flowers and forestry. In many cases, because New Zealand scientists team is more practically orientated, a lot of the work we're doing was actually world leading in those crops. They were the only, we were probably the first to generate in New Zealand, I would say, the first people in the world to generate the ability to genetically engineer white clover, ryegrass, kiwi fruit, tamarillo, asparagus, some of the flower crops, one of the first to do apples, and pine trees. So our scientists are actually leading a lot of this development around the world. We planted the first field trial in Lincoln in New Zealand in 1988. It was actually the first field trial in the Southern Hemisphere. And in fact, it was only one growing season behind the first field trial in North America and Europe. We're just one growing season out. And it was just the fact that our growing seasons vary, we weren't probably the first in many respects. But that was quite something. We were leading a lot of the work around field trials, evaluation, environmental safety, food safety of transgenic plants. 1988, there were just small potato lines, a small number of potato lines and a few asparagus lines were put in the field. Then over the next 15 years to about 2003, GE potatoes were field tested in most years out at Lincoln. They were usually small plots, less than 0.2 of a hectare. That's not much different than the size of this room. They were quite small plots, but in 1 year we got up to about a hectare. Now, it was really, really important we did those field trials when we did. Because they were a bit of a personal embarrassment, actually, those first field trials. We were growing these transgenic potato plants in the glass house, and they looked perfect. They looked just brilliant. We thought we'd done something really, really useful here. We could actually generate a really nice-looking potato plant with no changes from the original cultivar, except it was genetically engineered. And when we put the first field trial out in the field, it was quite a big occasion. Half the institute turned up to watch us plant them. There was television cameras, there was newspaper reporters, the radio were out there. It was a big day. And then as the season went on, it became really embarrassing. I'll come back to the slide shortly. I think I've covered most of that. Field trials, sorry, I jumped forward to myself. Field trials of GE crops in New Zealand. Overall history, there's been about 60 trials in New Zealand, mainly over the 20 years since 1988. Most of them are in Lincoln, most were potatoes, and they're about evaluating how good the system was, and I'll come back to that very shortly, selecting good lines and doing research on the impacts of those plants in an environmental way. Was the gene escaping into, how far was the gene moving in pollen? Was the gene escaping into microbes? What was the impact on the food products? But there's a list of all the crops that have been field tested in New Zealand. Quite a range of crops have been over the years, though nothing's been field tested for the last 5 or 6 years. Except there might still be some pine trees in Rotorua still growing. But this is our first field trial of potatoes. And you see there's some quite reasonable looking plants there. They are the controls that weren't genetically engineered. And you'll see there's some pretty scruffy looking plants back in here and in here. And some of them were so scruffy they didn't even survive the whole season. Others were so scruffy they looked more like black nightshade than potatoes. And look, this was a major embarrassment to me personally. But it was really important we found that out then at the start of the program. We could have gone on for another decade in the lab thinking we're doing great things. And this was not necessarily about genetic engineering. This is the fact that the plants were in cell culture. We always knew that when you grew plants in cell culture, from a single cell, they would often look abnormal. But we thought because they looked great in the grass house, they'd look great in the field. It's not the case at all. So we had to go back and reinvent our systems for engineering the plants, ensuring we could actually recover the plants much quicker. They had a very short period in cell culture, and from that we could generate really, really good plants. And it didn't take us too many more years to be able to grow really good potato field trials. Really good potatoes. This is actually a trial that got up to about 1 hectare. It was actually an example of herbicide resistance, and you can see some plots here. That are the control plots that the whole field's been sprayed, and this one's starting to die about 10 days after spraying, and you can see the control plots here, back there further, back there, and one more back there, of the control plots that are affected by the herbicide. All the other plots are growing just fine. So we were able to eventually generate really, really good plants. This is just another example of another trial in New Zealand, one of the other largest trials in New Zealand. This is canola. This was a seed increase done by for a Canadian company in the off-season of Canada. Sent the seed down, grow it here. That's quite commonly done for plant breeding. This was the first time it'd done for a transgenic crop. Grown under bird netting, that was done from our perspective to help stop things escaping, particularly by birds. But the company wanted the bird netting as well because these seeds are very, very valuable. They didn't want them eaten by birds. They were so valuable, those seeds. We had to recover every possible seed for them. But that was done for 1 year, and then we weren't allowed to do it anymore for Northern Hemisphere companies. The kind of things that have been achieved in New Zealand, I think the thing that I feel best about, that I managed to achieve, was resistance to potato tuber moth in potatoes. And by 2003, we had identified lines of potatoes in field trials that had really good resistance to that particular insect pest, and were as good as the controls in the plots as large as we could go, which were little plots of 5 rows of 10 plants, replicated about 5 times in a field, and we could detect no change in the plants morphologically or yield, except the fact that they were highly resistant to this insect pest. Disappointing to me that they never went any further. What's even probably more disappointing now is the cultivars that they were generated in, because of the success of plant breeding in the last 20 years, they're no longer growing. So the lines will be of no value now anyway, as it happens. If they could be grown now, they'll be of no value because we don't grow those cultivars anymore. Example of weed control in onions is the work of Colin Eadie out at Lincoln. Resistance to glyphosate or Roundup. You see the effects of when you spray a field once versus when you don't spray it. You can't even see the onions in that field when it wasn't sprayed. Another example is foliage colour in petunia. You see this lovely pigmentation gene in petunia. Quite a attractive in terms of its foliage colour, especially when planted with the green ones alongside it. I'll come back to that slide later. I want to make another point around that. That's a GM plant, field trialled in New Zealand. It's been done again in a different way. I'll come back to that later. And insect resistance in brassicas. Work of Mary Christie out at Lincoln, largely in forage brassicas, but also in cabbages, cauliflowers, Brussels sprouts. Against white butterfly caterpillar and diamondback moth. Brief comment about animal research in New Zealand. Has been done over the years on rats and mice, mainly as model systems because they're easy to handle and they're small. Mice, particular, and rats are always common model systems for human medicine studies, and they've been and continue to be used in New Zealand. Over the years, there have been projects on goats, sheep, and cattle in New Zealand, and a lot of that has led the world in terms of that technology. I was trying to recall the details, but I couldn't find them. But there was developed at Lincoln University in the early 90s a flock of goats producing a valuable protein in their milk. The idea is extract the protein from the milk. I couldn't find the details, but I do recall the flock getting up to about 100 individuals. All the current work is currently focused on a contained farm facility in ag research at Ruakura in Hamilton, where they largely do work on cattle, particularly dairy cattle, and sheep. Some of the things they're working on now, they've been able to remove by deleting the function of genes, allergens from cow's milk, and another project that they've been encouraged to work on is taking the black colour out of the cattle colour, because that's actually, in climate change in particular, caused a lot of heat stress in the animals being black, to reduce their heat stress. But that works I can't see it lasting politically much longer, other than the fact that there'll still be work on rats and mice as model systems to understand just as a science advance. No intention of releasing anything except Predator Free New Zealand is now funding some work in rats with the use of establishing gene drives, which you could release the rats potentially, that'll kill and diminish rat populations. So that is work that's underway at the moment. Wanted to focus briefly on introgenics and gene editing. Now these are sort of softer versions of genetic engineering because no foreign DNA ends up in the resulting plants. That's the concept behind them. And all the work up there I've described, we're putting essentially foreign genes into plants. Often they're genes or DNA from bacteria into plants, become part of their genome. And the vectors we use to carry those genes into the plant cells are all based on bacterial DNA. So even if you're putting plant genes, or even if you're putting a potato gene back into a potato, the vector you've used to carry the gene is built on bacterial DNA. So the transgenic plant ends up with bacterial DNA plus the gene you want. Now the concept behind introgenics is you build your vector out of the plant's DNA. And it's something that we actually developed first out at Lincoln, where we could actually identify the fragments we needed to build the vector from within the genomes of a range of plants, and you'd use those fragments to build your vector, and then you'd put genes from that particular plant on that vector and transfer them between cultivars. In many respects, because no foreign DNA ends up in the resulting plant, it's all from the plant itself or the gene pool available to plant breeders, the outcomes are identical to well-established methods that I described early on, particularly around mutation breeding and radiation-induced mutations. You're just moving things around the genome that are already there. Now these concepts, introgenics and gene editing, they create a bit of a dilemma because under New Zealand definitions in the Has No Act, they are still considered to be GE plants. That happens in New Zealand and also in Europe. But aspects of these technologies in other countries are not regulated, so the plants are not GM. and this is going to cause a dilemma in about, I reckon, within the next decade, is that in North America and to some extent in Australia, because gene editing is not regulated in aspects of gene editing are not regulated in Australia, people will develop new plants, they'll become part of the breeding programs, and seed gets moved around the world all the time, every year moved around. And before you know it, well, we won't know it at the time, but we're growing these kind of crops, and in hindsight we'll find out later, oops, they've been done through introgenics or gene editing. I can see that being a dilemma politically moving forward in the next decade. So the term of introgenics, all the DNA transferred to the plant originates from the target crop. This includes the genes to be transferred, as I said, and the way we viewed it was, as long as you took the genes from the gene pool that a plant breeder could access through traditional breeding, you could use those genes and still call it introgenics. And we built the vector out of the host plant's DNA as well. So we end up with these sort of non-transgenic GE crops, and I said they were really identical to microtranslocations that could result from radiation-induced mutations. Now I'm going to show you one more slide to illustrate this. This is how we often illustrate the genomes of plants once we've got the whole genome sequenced. This actually represents the 24 chromosomes in a tobacco plant, just used as a model system. So each little stretch is the DNA in a particular chromosome, range from chromosome 1 round to chromosome 24. This here is a diagrammatic view of the vector used to carry or transform DNA. Whoops. Big thumbs. Sorry. This little stretch here represents the vector. So we've built this vector out of the tobacco DNA. So there's actually, the vector composed 4 fragments, numbers 1 and number 2, number 3 and number 4. And number 4 came from chromosome 17, the fragment from 3 came from chromosome 19, 1 from 21, and 2 from chromosome 4. And on that we put this gene. This is a gene for this case, herbicide resistance. And it came from, unfortunately, a small region that was yet to be allocated to a chromosome, but despite it is tobacco DNA. So we built the vector and then went through our usual procedures and engineered tobacco. And once we recovered the plant with herbicide resistance and sequenced the genome of the recovered plant, we found that the gene had now been inserted into chromosome 5 at this location and only composed of a small part of fragment 2, the gene, and a small part of fragment 3. So the resulting plant was only a minor rearrangement of the tobacco genome, much like you'd expect through a mutation breeding program. And I showed you this photo before, the Petunia plant, the GM field trial in New Zealand. The people, scientists in Palmerston North, Kevin Davies and colleagues, and Murray Bowes, remade this plant using an introgenic approach. Using only Petunia DNA, remade that coloured plant. That plant is now grown in the USA. It is not regulated. It did not trigger the definition of GM, and that plant is now grown in the USA. Wouldn't surprise me that it comes back through some horticultural company as a cultivar back in New Zealand at some point in the future, and people not know about it. You might well ask, if all you're doing is moving plants within a species that could be moved by plant breeding, why don't you just use plant breeding rather than this technology? Which is a fair question. But really, to me, the real key thing is what we can achieve through this approach is direct gene transfer into the elite lines of plant breeders in a single step without backcrossing. So if I wanted to, through plant breeding, transfer a gene from a wild potato species into potato through genetic engineering, through plant breeding, it would take me 10, 15 generations at least. That's at least 10 to 15 years. Whereas I could use this technology and do it in about 6 months. So it's much more efficient. And in responding, to me, the climate change crisis, climate change is one issue. It's actually having food to grow under the new climates, it's going to be critical as well. And there's more pressure on our breeding programs to produce adapted crops to the changing climate and the changing disease spectrums induced by that climate change are going to be a real issue in the next 10, 15 years. So I see that as being a potential way forward to speed up the breeding program. And the other key advantage is you can simply transfer just the gene of interest and no other baggage that comes with it that we call linkage drag. And I'll come back to that a bit later on. You're just transferring the gene of interest and not other genes as well. Gene editing is a new technology. Been around for about 10 years, and now can be achieved in almost every crop plant. It's the latest technology for inducing very small changes for breeding purposes in a plant. You can target a single base pair of DNA and change it. And because we now know for most of our crop plants the full genome sequence and all the genes, you might say, that gene, I want to create that particular mutation to cause that effect in the plant. And you can target now, and you can quite simply do it. It sounds quite manipulative and scary, as though you're controlling life, just like all GM things, but you need to put it in context of other approaches, and I'll come back to that shortly. But of all the genetic approaches used in plant breeding throughout history, gene editing is the most highly targeted, causing the least change in a genome compared to all the other technologies. How does it work? There's a range of processes, but the most commonly used is what we call the CRISPR-Cas9. And that's an enzyme complex that binds DNA, and you can target it by using an RNA molecule with the sequence of your target gene on that RNA molecule. And this enzyme, the CRISPR-Cas9 complex, will then find that in the genome, and it'll cut the DNA at an exact site that you want to cut it. And then you rely on DNA repair mechanisms to repair that DNA that all plants and all organisms have, and you look for the mutation that you might want to cause at that particular site. That's going to become a very common way of going about things in the next decade, especially now that we have the whole genome. We know them for most of our crop plants already, and we'll know them for the remainder crop plants in the very near future. And I want to illustrate this with one more example. This is a forage brassica here, and this is grown in New Zealand, and I would say almost all the forage brassicas you see now growing in New Zealand have this particular mutation in them. The mutation has been induced by chemical mutagenesis to recover a herbicide-resistant plant. The herbicide is, in this case, Talar. It's what we call a sulfonylurea herbicide, and that herbicide targets the AHAS gene, acetohydroxy acid synthase gene. It binds to the enzyme, think back to this first slide, DNA, RNA, protein. The protein or the enzyme from the DNA, from the gene, that herbicide binds to the enzyme and prevents the enzyme undergoing its usual function. But in a resistant plant, we found that the resistance was due to a single nucleotide change. So DNA is read in what we call codons. Codons are 3 nucleotides. So rather than having CCT, the resistant plant had TCT in its DNA, and that led to 1 amino acid changing in the protein. It resulted from, rather than a proline, you got a serine. And that small change of amino acid in the protein was enough to cause a slight conformational change in the enzyme that prevented the herbicide binding to the enzyme. Therefore, the enzyme could continue to function in the presence of the herbicide, and the plant is resistant. That was done through mutation breeding, through a chemical known as EMS, ethyl methyl sulfonate, a really nasty chemical because it causes mutations. But of course the EMS is no longer present in the crop, and that crop is now grown largely throughout New Zealand in forage brassicas. And you can see the value of it in terms of herbicide resistance there in the picture. You can see the field that's been part of the field that sprayed and part that wasn't. Reduction in weeds more production for the farmers, and what's more importantly, the weeds aren't growing, therefore the animals are not eating the weeds. The weeds often bring in things that taint the milk and the meat. That mutation, the identical mutation, could be done through gene editing very, very easily, quite quickly. From the original herbicide-resistant plant, it took 15 years to release the first cultivar. It took that long to backcross that gene into good cultivars. You could come in through gene editing and do that in about 6 months, I reckon. So that's the value of gene editing in the future. And the frustrating thing as a plant scientist, you could do it in a more efficient way, very quickly, but it's regulated and you can't do it legally, release it, but it's already out there. Right, just wrapping up. I showed you this order before. The history of plant breeding. As you come down that list from early civilization to gene editing, there's a decreasing disruption of the genome. So the modern technologies disrupt the genome less. And there's increasing precision of the genetic change you're making. And I think that's one of the important take-home messages I'd like to leave with you. Looking at gene transfer by traditional breeding. This has been done for many decades. Plant breeders are often cross between species. They'll bring in genes for disease resistance from a wild species into a crop plant. Undefined genes are routinely transferred between breeding lines, between species, and in some cases between different genera of plants. It's often associated with large chromosome fragments from the wild species ending up in our crop plants. Only a small fraction of that transferred DNA carries the gene of interest. The rest of it is what we call the linkage drag, other genes that come along with it. And also importantly, often when we transfer genes between species through traditional plant breeding, we don't know their function. All we know is they give an effect, disease resistance. We don't know how that resistance is derived. Is it making a toxin to the disease? Is it doing something else? We've got no idea. Yet we accept that, and we have done for decades. Got a lovely example here from Richard Pickering, who used to work out at Lincoln. And Richard, over about 15 years, was able to transfer powdery mildew resistance from Hordeum borbosum, a wild species of barley, into cultivated barley. More impressively, he was able to do the cytology and differential staining of the genomes in his resulting plants. So this is the chromosome spread of the 14 barley chromosomes, the 7 pairs of barley chromosomes of the cultivar resistant to powdery mildew. The little glowing part there on the tip of a pair of chromosomes represents the DNA he transferred from Hordeum borbosum that's now in cultivated barley. That's where the gene for powdery mildew resistance is located. Now we don't know how that resistance is caused. It might be a toxin to powdery mildew, we don't know. But we accept that technology. More importantly, if you imagine that little dot, coloured dot there, is probably about 1% of the whole chromosome base. Barley has 30,000 genes. 1% is 3,000 genes. There's probably 3,000 genes on there from Hordeum borbosum. We don't know what all the others do either. But we accept that as opposed to what we can achieve through genetic engineering. In the case of a genetically engineered crop, every base pair you're transferring, we know what it is. We can work out after the fact exactly where it's gone. The RNA and the DNA, the RNA and the protein that's made from the gene, we know what they are. They're well understood. They had to be to get the gene in the first place. We have sensitive assays to measure the presence of the DNA, the RNA and the proteins. We can work out exactly where those modified proteins are in the plant and where the RNA is and how much there is. So the environmental impacts and food safety issues of genetically engineered plants are a consequence of the phenotype or what comes out, what the final plant is, not its genetic status, in my opinion. It's not about how you derive the plant, it's the effect it has. And I want to finish really with two statements that I've been building up to. I wrote these over 20 years ago. I think it was in the late 80s, early 90s, when I was part of a FAO and World Health Organization panel on safety of GM crops. And I don't think I want to change a word nowadays. I have every confidence in those then, and I do now, that the risk of GE crops to natural ecosystems, agricultural ecosystems, food industries and consumers will be no different than the effects of growing, processing and eating cultivars from traditional breeding. And I'll go a step further. The precision and power of molecular biology offer greater confidence of achieving and monitoring the desired outcome in comparison to traditional plant breeding, because we have the knowledge base is that much greater. So really that's my take-home message, and I've got one more slide, just to reflect back on history again. Thomas Fairchild, a nurseryman working near London in the 1700s, he's credited with making the first hybrid between species. He was able to cross Sweet William and Carnation. And he was subjected to many accusations of blasphemy at the time for tinkering with God's creation. And to think what we've achieved in plant breeding since then is quite amazing. But another important quote from Luther Burbank back in 1906, when Mendel's laws of inheritance were first rediscovered. He wrote, "We have recently advanced our knowledge of genetics to a point where we can manipulate life in a way never intended by nature. We must proceed with the utmost caution and application of this newfound knowledge." And that's written almost 120 years ago. And genetics has gone a long way in that time. Yeah. Thank you very much.
Well, Dr. Connor has invited questions from the floor. Anyone like to ask a question? There's someone around the back. And if you give your name, please.
Kia ora, Peter Gordon. Why is it that genetic engineering is not allowed?
Public pressure, primarily, and political reasons, I guess. Because there's been a lot of debate. I think one of the frustrating things about it is that because it's become so politicised, one of the criticisms is all controlling plant germplasm by big multinational companies. And look, that is a fair criticism. All the crops are owned by the big multinational companies. But the system is so rigid and so complex and so expensive to abide by, it's driven to the point where only the big multinational companies can play in that space. I could do this kind of technology on my kitchen bench at home. With the knowledge I have, it wouldn't be difficult to actually engineer crop plants in the kitchen at home. Small companies can play with it, but they can't afford to play in it because the regulatory burden is so high. And to me, that's the problem, is that the big companies know that and they drive the system to be that way. Whereas if we freed it up, smaller players could take part and we could make a lot more advance in our plant genetics, in my opinion.
So has there been a political approach made by people such as yourself?
I have got very tired of making such approaches. Yes, look, the last national government promised to do something every time they came to power. It never happened. The Labour government, I should be careful what I say here, they would like to make some changes, but they have this coalition partner which they rely on that it's not appropriate at the moment for them to do so. That's what I understand. But it needs a change in the Has No Act to free up the ability to do things.
Hi, yes. My name's Gary Adams. I'm an ex-farmer. I enjoyed your talk very much. I don't disagree with anything much you've said, but my question is a little bit outside of tonight's subject in a way. A very, very big topic at the moment, of course, is nitrates in Canterbury on the dairy farms, nitrate poisoning in the water, that sort of thing. As a scientist from Lincoln, have you got any thoughts on that at all, please? Pretty big subject.
Big different subject, but yes, it's a major problem. I accept that. It's just the intensification of agriculture has caused it. The change in farming systems, particularly dairying in Canterbury, have been a major problem. While you can make a profit on something, people will make a profit on something. I won't try and defend it because I don't agree with it. I think we should come up with a more sensible solution to the way we go about our farming. And to be honest, a lot of farmers are trying. They're trying really hard, but it's a balance of being able to produce a living for the farm as well. But it's about what they farm and where they farm is where I have my concerns. And we need to think about some land use changes in agriculture.
Ivor Watson. I have two questions. One, you made a reference about rats and how a change in genetics of the rat could reduce the rat population. Is there any work going on of a similar vein with opossums? Because as you know, they are a major concern in New Zealand. That's one question. The other question is, you mentioned about the CRISPR-9 being able to cut the DNA at a very specific point. What I didn't understand is how does that, the DNA will repair that cut, but how do you actually control it so that you have inserted what you really want into that cut?
So I'll deal with the second part first. Two ways. You can actually target and engineer yourself the actual insertion of the change, but that becomes a little bit genetic engineering again when you insert something back in, or you can rely on nature to repair it. You reduce your plants and you search amongst the plants for the mutation you want. Is the other way of going about it, relying on natural repair. And the earlier question about the rats and the possums, yes, applies to possums too, but of course there's also a risk around. It's a theoretical concept that you could engineer an organism with a deleterious gene that when it becomes homozygous, the individual dies. So you release what we call heterozygous individuals. They go out in the population, and by using what they call these gene drives means that you can establish ways in which you bias or increase the frequency of recovering homozygous, the way you control the genetics of the situation. So you increase the number of homozygotes and then they die, so the population declines. It is largely a theoretical concept at this stage, but I am sure smart people will find ways of achieving that, and it could be applied to any organism. But the risk around it again is, well, we do have our native Karori rats. Would it get into them? What if the possum got back into Australia where it's actually an endangered species? So it creates a lot of political issues as well, of whether you should do it or not. So it comes down to the ethics of science.
Thank you. I'll ask Jane Weber too.
Kia ora Tony, ko Jeanette Weber aho. Thank you for your really excellent presentation. It has demonstrated exactly why we come to U3A. You have shared your knowledge, your experiences. They're all very extremely interesting and very, very clearly explained. we have experienced a great deal of new learning and a deeper understanding and appreciation of genetic engineering in agriculture. This is a small koha. Thank you, please accept it.
Thank you.
Yeah, thank you.
Thank you.
Kia ora, kia ora.
Thank you very much. Well done.